My name is John Chappell. I'm an associate professor at the Fralin Biomedical Research Institute at Virginia Tech in Carilion. And my lab works on vascular biology. So we try to understand the basic mechanisms of how blood vessels, particularly endothelial cells and a cell type known as a pericyte, work together to form new vessels in the developmental context. We also try to understand how blood vessels remodel themselves in different diseases as well. So our interest in studying blood vessels in von Hippel-Lindau disease scenarios is trying to understand how the genetic mutations that give rise to the disease state also affect the blood vessels in different tissues. And particularly, we're interested in the eye as well as the kidney. And recently, we found that in the kidney, that these vessels undergo some early transformations after the genetic mutations are induced in a preclinical animal model. And so this work is supported by the von Hippel-Lindau Alliance. And we're excited to find some of the early remodeling events and the mechanisms that underlie them might be relevant to clinical targeting to potentially slow them down or perhaps cause them to stop altogether and prevent downstream disease scenarios like clear cell renal carcinoma, which is highly prevalent in VHL populations. And so we're excited to study the von Hippel-Lindau disease scenarios so we can fit a piece into the whole puzzle in developing new clinical therapies that can help VHL patients, particularly those that might be able to target the vascular system, slow some of these changes from occurring, perhaps stop them altogether would be ideal, and especially prevent some of the downstream formation of the clear cell renal carcinoma or hemangioblastoma formation. So we're excited to contribute to the community of VHL research because it's essential for our research to partner with those clinical studies as well as patient insight to understand how we can, as a community, treat this devastating disease. All right, let me think. So in particular, one of the interesting findings that has been emerging from my lab is through a team effort. One of my graduate students, Zuzka Mironovova, has been working on this preclinical model of von Hippel-Lindau disease. And in particular, she independently found that these changes in the kidney are manifested in changes in the extracellular matrix surrounding blood vessels as well as the kidney glomeruli. And so this was an unexpected finding, actually, that after inducing these mutations, that these changes in the structural basis of the vessels in the kidney actually happen so quickly. And also, interestingly, these mutations induce signatures that are found in the human population, including increased red blood cell count, which is found in von Hippel-Lindau patients in Asia. So Zuzka's work is helping us understand how these changes might occur and the molecular underpinnings for how they are affecting the kidneys.